Hello everyone, Mr. Linder here. Let's continue talking about transport processes. So in the previous videos, I was talking about some of the difficulties that we have getting substrates, ligands, substances across the plasma membrane. And some of the difficulty lies in that not everything is lipid soluble. And so when you have molecules and ions that can't just directly cross the plasma membrane, we need specific mechanisms to get those substances across. So I want to continue talking about this idea of carrier proteins. So carrier proteins are proteins that bind to specific substances, or again, we can call them ligands. What we're really talking about are molecules, ions, and assisting those molecules or ions to get across the plasma membrane. So in this diagram, we're looking at different types of carrier proteins. For example, this carrier protein is moving glucose, so that would be the substrate, again, or the ligand, and it's moving that glucose across the membrane. Here we have a carrier protein that's moving both glucose and sodium across the plasma membrane. Here we have a carrier protein moving sodium across the plasma membrane in one direction and potassium across the plasma membrane in another direction. So we have different types of carrier proteins that are going to be involved in moving molecules, ions, etc. Across the plasma membrane. So we can categorize these carrier proteins. If it's a uniport carrier, uni means one. So here in this example, this is one glucose. So that one glucose moving across the plasma membrane, we're talking about a uniport carrier. So that's a uniport carrier protein moving glucose across the membrane. A good example of this are the GLUT transporters, GLUT. We'll talk more about these, but glucose transporter, GLUT, they may have a number associated with them, so it might be a GLUT2 or a GLUT5. Those numbers just represent locations within the human body. So as long as it says GLUT, then we know it's a glucose transporter. And since it's moving one molecule, it's a uniport. Co-transporters are going to be carrier proteins that move multiple molecules or ions or some combination across the plasma membrane. So here we have sodium and we have glucose, so multiple items moving across the membrane. So this is a co-transporter. Here we have sodium and potassium moving across the membrane through this carrier protein. So again, this is some form of co-transport. If the molecules and ions are moving in the same direction across the plasma membrane, we say that this is a symport carrier. So moving in the same direction is a symport carrier. If they move in opposite directions, so here we have sodium going one way and we have potassium going the other way, this is an antiport carrier. What you'll also notice is that you can have carrier proteins that utilize energy. So if there's ATP involved in the process, we know it would be active transport because active transport requires energy. If there's no ATP involved, then this would be passive transport. So just in this diagram, we have examples of passive carrier proteins, and then we also have an example of an active carrier protein. So when you start describing proteins in the membrane and what they actually do, we get into a lot of different definitions of things. So is it a carrier protein performing symport? Is it a carrier protein performing antiport? Is it using passive transport? Is it using active transport? So there's a lot of definitions that we can use. When you're using a carrier protein, because there has to be interaction with the molecule or ion with the protein, the transport is going to be slower than just using a channel protein. Another thing to consider, so in this diagram, another thing to consider is that the Carrier protein does not create a continuous pore or hole from the extracellular to the intracellular fluid. And that's different than what you see with your channel type proteins. If we just had a channel protein, we can see that there can be a continuous passageway from extracellular fluid to intracellular fluid. But with carrier proteins, you'll notice that it's only open to one side at a time. It's similar to this idea of a lock system like we see in, say, the Panama Canal, where you allow water into one section of the lock. Ships can pass in. You have to then close. So we have an open situation here. Then you have to close that side of the lock, and then you have to open that lock to allow water to flow to the opposite side. That's similar with this carrier protein concept. So here we have a molecule binding to the carrier protein, and then it's open on this side, but it's still closed on the intracellular side. There's a conformational shape change that takes place in the protein, and so now it's open to the intracellular side and closed off to the extracellular side. So there's no continuous passageway between extracellular and intracellular. And so that's a difference we see between channel proteins and carrier proteins. Facilitated diffusion is a great example of using this concept of a carrier protein. So back to the glut transporter. So the glut transporter is a good example of facilitated diffusion. And so what it's doing is it's binding glucose on one side of the membrane, in this case the extracellular fluid side. It's binding glucose, it's doing that conformational shape change, and then it's allowing that glucose to enter into the intracellular fluid. So we're using a concentration gradient, we're moving glucose from high to low, so this is an example of diffusion, but it's facilitated diffusion because it requires that carrier protein to interact with the glucose and then open to the inside and allow the glucose to move in. So facilitated diffusion may look like simple diffusion, but it's really not. You're utilizing a carrier protein. You have conformational shape changes that take place in order to move that molecule or ion across the plasma membrane. So if you give a definition of facilitated diffusion, it's a passive mechanism that moves molecules or ions from a high concentration to a low concentration, but it utilizes a carrier protein to perform that task. And so these glut transporters are a great example of facilitated diffusion in action. Now, what's interesting is that in order to maintain the concentration gradient and not reach equilibrium, because we know that with diffusion, if a molecule moves from high concentration to low concentration, eventually the particles inside the cell will increase in concentration as the particles outside the cell will decrease. And at some point, the concentration outside will be equal to the concentration inside. And so you would end up with an equilibrium situation. To avoid equilibrium and to continue glucose moving into a cell, what your cells will do is we will either take the glucose and turn that glucose into energy. So you can immediately take glucose that comes into the cell and run cellular metabolism, so cellular respiration. And by doing that, you can produce ATP. So we can perform processes like glycolysis, the transition reaction, the Krebs cycle, the electron transport chain. And you can take that glucose that has entered into the cell and you can turn it into ATP. And if you're doing that, that's going to keep the concentration of glucose low inside of the cell. Another thing that cells do is that we turn the glucose into something else. So when glucose enters into the cell, we can produce something called glucose 6-phosphate. And glucose 6-phosphate is actually a molecule that's used in glycolysis, but it also can be converted into a storage molecule called glycogen. So we can actually spend some energy to produce glucose 6-phosphate within the cell, and that's going to keep the concentration of glucose low inside of the cell. And that keeps that high to low concentration gradient so that we can continue to have facilitated diffusion into the cell. So it's a way for us to maintain that facilitated diffusion without reaching equilibrium. The next thing I want to talk about is active transport. So active transport is where we move molecules or ions against a concentration gradient. And in order to do that, there has to be an input of energy. So active transport requires energy in order to move molecules against their concentration gradient. And what this does for us is it actually creates this idea of disequilibrium within the body. If you were to allow, let's say, for example, if you were to allow sodium to diffuse from outside of our cells to the inside of our cells, they would eventually reach equilibrium. Those ions would reach equilibrium. But if you have active transport, so let's say we have a carrier protein that uses ATP that can move sodium back out of the cell, you are creating disequilibrium. You're spending energy to ensure that you keep a high concentration of sodium outside and a low concentration of sodium inside of the cell. So oftentimes think of active transport as moving things against the concentration gradient so that we don't have equilibrium occur within the body. Active transport is divided into primary active transport and secondary active transport. If it's primary active transport, it uses ATP directly. So you'll see the carrier protein utilizing ATP energy to perform its transport action. The sodium-potassium pump is a great example of a primary active transporter. Calcium pumps are a great example of primary active transporters. So these are all primary active transporters on this table, but they utilize ATP directly. So in this diagram, this is a calcium pump. So what we have here is we have this carrier protein that's going to interact with calcium ions. The carrier protein must utilize ATP in order to be functional. So it's active transport. And what kind of active transport is it? It's primary active transport because the energy is being utilized directly by the carrier protein. And what this carrier protein does is it pumps calcium from inside of the cell to the outside of the cell. And so we have calcium moving from one area of the cell, intracellular fluid, to the extracellular fluid. And it's doing it against the concentration gradient. So we're actually moving low levels of calcium to high levels of calcium outside. So that's why it requires energy, because you're going against that concentration gradient. We're moving low to high instead of high to low. The other great example is this sodium-potassium pump. Here we have a carrier protein that is using ATP directly. So this is a primary active transporter. It's pumping sodium out of the cell, and it's pumping potassium into the cell. And that's going against the concentration gradients. Cells have low sodium on the inside, high sodium on the outside. So if you're pumping sodium out, it's against the concentration gradient. Cells have low potassium on the outside, high potassium on the inside. So again, you're pumping against that concentration gradient. And so the only way you're going to be able to do that is by utilizing ATP. Now, how many ions are actually moving? So we can take a look at this diagram. You'll see it's a carrier protein. And again, carrier proteins have to go through those conformational shape changes. So here we see 3 sodiums binding to the carrier protein. So there are 3 sodiums that are going to be moving out of the cell. And then we have 2 potassiums that are going to bind to the carrier protein, and those potassiums are going to come into the cell. So it's 3 sodiums out for every 2 potassiums in for the sodium-potassium pump. And this sodium-potassium pump is utilized all over the human body. And again, it helps us to maintain that concentration difference with sodium and potassium, that disequilibrium that we can use in other physiological mechanisms, and we're able to do work with that. The secondary active transport is a form of active transport that relies on energy from the primary active transporter to then power another protein within the system. And I'll show you an example of that. So secondary active transport is actually using the potential energy that is created by a concentration gradient that came from the action of a primary active transporter. What you'll notice from this particular table is that a lot of these secondary active transporters are sodium dependent. So using the sodium concentration gradient, which is maintained by the sodium potassium pump, remember the sodium potassium pump uses ATP to create that or maintain that high level of sodium outside of cells. And so we have high sodium outside, we have low sodium inside. The only way that's maintained is to use the sodium potassium pump to move that sodium out of the cell. Right? Sodium out, potassium in. And so we're using ATP to do that. Once you have a high concentration of sodium, you can link that with the transport of other molecules or ions. So here we have sodium linked with potassium and chlorine transport, sodium with glucose transport, sodium with chlorine transport, sodium with bicarbonate transport, sodium with some form of amino acid, a glycine, an aspartic acid, etc. Okay? Sodium-dependent transporters, okay, fall into this secondary active transport, where we utilize energy from the primary transporter to run a secondary transporter. Now, that doesn't mean there aren't other types of secondary active transporters. It's just that sodium-dependent ones are the most common. We do have other, let's say, hydrogen-dependent ones. Okay? So there are going to be examples. What I want to show you in this diagram is kind of how this secondary active transport would be set up. So the primary active transporter, the sodium-potassium pump, is creating a potential energy situation. By pumping sodium out of the cell, you are creating that high concentration of sodium outside of the cell. And then sodium can diffuse, right? Because of the high concentration, it can diffuse along its concentration gradient, and it can power the movement of something else. So in this example, sodium is moving up this block, it's gaining potential energy, sodium falls off the block, hits this teeter-totter, and it moves glucose. Okay? That glucose, though, doesn't move without the sodium. And the sodium doesn't move unless you have the sodium-potassium pump spending energy using ATP to maintain that concentration gradient outside of the cell. So this is a form of secondary active transport. The teeter-totter is not using the ATP directly, but it's benefiting from the use of energy to create the potential energy of sodium outside of the cell. And so that's why it's referred to as secondary active transport. It's somehow tied to the use of an active transporter somewhere else. You can have beyond secondary ones too. So we start with the primary to get to secondary, but you can have tertiary level transporters as well, or quaternary level transporters, as long as they're tied to that first transport, the utilization of ATP, and then all of these other transporters work together. You can have secondary, tertiary, quaternary level transporters. A great example of secondary active transport is the sodium glucose transporter. So we saw earlier that you can just have a glut transporter, but a glut transporter is facilitated diffusion. That's just using the high to low concentration gradient. It's just a form of passive transport using the high to low gradient. This sodium glucose transporter is dependent upon the primary active transport that's created by the sodium potassium pump. So what's not shown in this diagram is the fact that you have a sodium potassium pump that has created the high concentration of sodium outside of the cell. So where does that high concentration of sodium come from? It comes from the primary active transporter. This carrier protein in blue then becomes the secondary transport. And what we see here is sodium binding to the carrier protein, and when it does that, it allows for a conformational shape change so that now glucose can bind to that same carrier protein. And then both sodium and glucose can enter into the cell. So this would be a symport, using our vocabulary. This is a symport, moving sodium and glucose in the same direction. It's utilizing a carrier protein. But what form of transport is it? This is secondary active transport. And again, the reason it's secondary active transport is because you are utilizing the energy from the primary transporter to maintain that high concentration of sodium outside of the cell to then power the movement of the glucose. So a glut transporter is facilitated diffusion, but an SGLT, a sodium glucose transporter, is secondary active transport. So take a look at that example and make sure that you understand how it's different from facilitated diffusion and how we have secondary active transport. So the sodium-dependent transporter is a very important concept for physiology. When we look at carrier proteins and carrier-mediated transport, something else to consider is their specificity, their competition, and these important concepts. So when we look at, let me go back to this diagram, when we look at this idea of specificity, we can have carrier proteins that are specific for the transport of just one molecule or just one ion. So the example was the glut transporter. Glut transporters are specific for just hexosugars like glucose. They may also be able to move galactose or some other hexosugar, but they cannot move something like maltose. So a glut transporter only moves, say, glucose, but it can't move maltose. So in this example here, maltose, which is a disaccharide, when it tries to bind into the carrier protein, it's not able to move across the plasma membrane because there's specificity to this particular carrier protein. Instead, what we end up having is competition. Maltose acts as a competitive inhibitor. And therefore, once maltose binds to this carrier protein, it would be inhibiting the movement of glucose across the membrane because glucose now can't fit into and bind with that carrier protein. So competitive inhibition can actually slow down or stop the transport of a particular molecule. Now, how can we use that medically? Well, clinically or medically, we use oftentimes medications to help benefit patients. For example, there are medications to treat a condition called gout. Gout essentially is caused by increased uric acid levels. So when your uric acid levels become elevated, uric acid can lead to arthritic conditions and therefore pain in your joints. And so we have to find a way to lower those uric acid levels in the body. And so what these gout medications do is they competitively inhibit the transporters within the kidneys. So if the kidneys would be reabsorbing. Let's say that, for example, we're looking at a cell in the kidney, and we're looking at the blood, and we're looking at these cells lining what's called the nephron. Okay, so the nephron is a tube. It's a series of tubes within the kidneys that we use in the process of filtration and reabsorption and secretion. And we use this nephron system and the cells that line the nephron to perform the physiological functions for producing urine. So let's say we have these cells, though, that would be taking uric acid from the nephron and reabsorbing it back into the bloodstream. Well, if those cells were doing that and you had excessive amounts of uric acid, that would be increasing your uric acid in the blood. So what if you took a medication, so we'll put MED here, what if you had a medication that comes along and competitively inhibits the carrier protein, so we get a medication here that blocks the carrier protein and doesn't allow the uric acid to be reabsorbed back into your bloodstream. That would mean that the uric acid would keep going through the nephron and eventually be eliminated from the body. And so it would collect eventually in your urinary bladder, and you would eventually eliminate that uric acid from the body. So we can use competitive inhibition as a way to control, say, uric acid levels and treat somebody with gout. Another thing to consider about carrier proteins is that carrier proteins can become saturated. So there are diagrams that we can take a look at just to show you that carrier proteins have a maximum rate of transport. When a carrier protein is maxed out, meaning all the carrier proteins are being used by a particular cell, you can only transport up to that maximum. So if you have a cell that is limited in number with carrier proteins, just for simple examples here, let's say we only have, just kind of drawing in some things here. Let's say we only have 4 carrier proteins. When all 4 of those carrier proteins are being utilized and something is being transported across the membrane, we've reached a maximum rate of transport. When there's only 1 or 2 molecules to move, we would still have a couple extra carrier proteins available. And so if more substrate was available, we could move more of those substrates across the membrane. But your cells are limited by the number of carrier proteins that they have. So what we see in this diagram or in this graph is that as the concentration of substrate goes up, there will be an increased rate in transport until you reach the saturation point. Once you have utilized all of the carrier proteins that are available, so these are all the proteins that the cell has, then you can't go any faster. You reach a plateau. So it doesn't matter if we keep increasing the concentration of the molecule that we want to move across the membrane, we can't transport any more of that molecule. In order for a cell to increase its rate of transport, the cell would have to manufacture more carrier proteins. So if we were able to put more carrier proteins in the membrane of the cell, then we could increase that rate of transport. But eventually it would plateau again, right? Eventually you would reach a transport maximum again when you utilized all those new carrier proteins. So those are concepts that you should understand when you're looking at transport processes. The last thing to take a look at is vesicular transport. In vesicular transport, we're talking about active transport, but we're talking about moving larger particles. We're talking about moving sometimes entire organisms across the plasma membrane. In vesicular transport, we're looking at things like phagocytosis, which is basically translates as cellular eating. We're looking at things like endocytosis, to bring these molecules or particles into the cell in a very specific way. So, and then we're looking at exocytosis. All of these mechanisms require ATP. So in phagocytosis, think of it as a mechanism of cellular eating. So when you need to engulf a bacteria, as shown in this diagram, or you need to engulf large macromolecules, you are going to use these pseudopod structures. So the pseudopods are basically the extension of the plasma membrane, seen here. These are pseudopod extensions. And what's happening is we're using actin proteins, these basically contractile type proteins, to move the membrane around what you're trying to engulf. And so the pseudopods continue to extend around this bacterium until you put it into what's called a phagosome. The phagosome then pinches away, so you can now see the phagosome is inside of the cell. And then phagosomes can merge with lysosomes. Those are shown in green here on the diagram. And lysosomes contain digestive enzymes. And those digestive enzymes can be used to break down what has been brought into the cell. In this case, a bacterium, but it could have been a large protein. And so you're using digestive enzymes to break down that protein. White blood cells use phagocytosis on a regular basis to pick up foreign particles, to engulf bacteria, and it's part of the immune system. Endocytosis, phagocytosis and endocytosis are clearly similar to each other, but endocytosis typically is used to describe bringing in smaller molecules or ions, and it's usually creating these much smaller vesicles. So when we look at the concept of endocytosis, we want to think of the membrane sort of pinching in. So here we have this pinching in that's taking place, and you're creating a smaller vesicle. So that would be this endocytosis. Endocytosis can be specific, but it can also be non-specific. We use the term penocytosis to mean cellular drinking. That's typically a non-specific type mechanism of bringing molecules or ions into the cell. If we use the term receptor-mediated endocytosis, we're talking about bringing specific molecules into the cell. These processes, though, require ATP. Again, if it's non-selective, it's penocytosis. If it's highly selective, it's receptor-mediated endocytosis. Penocytosis translates as cellular drinking, and again is non-selective. But receptor-mediated endocytosis utilizes receptors. So in this diagram, there are receptors depicted in the plasma membrane. And this receptor is going to interact with a specific molecule or ion in the extracellular fluid. So that receptor is going to bind to that ligand. Again, a ligand is just a term we use, just like we can say a substance. Okay? It represents a molecule or an ion that's going to interact with the receptor. So some ligands might include antibodies. They might include cholesterol. They might include iron, growth factors, hormones, all kinds of different ligands that can specifically be brought into a cell using receptor-mediated endocytosis. And so how does this receptor-mediated endocytosis process work? We use what's called the clathrin-coated mechanism. So in this diagram, you'll notice these clathrin proteins that are embedded into the plasma membrane. And this is where the receptor and the ligand are going to migrate to. So the receptor and the ligand, once they've interacted with each other, they're going to migrate to this clathrin-coated pit area. So ligands are going to bind receptors, and then the receptor and the ligand are going to move to the clathrin-coated pit area of the plasma membrane. Then the membrane is going to start to pinch inwards. And so we see here this pinching in of the plasma membrane. And that is what we refer to as endocytosis, bringing something into the cell. Eventually, the vesicles are going to pinch away from the membrane. And then once they pinch away from the membrane, you'll notice that the clathrin proteins are being recycled back to the plasma membrane. So we're losing those little clathrin proteins, and you're left with a vesicle that has the receptor and the ligand inside of it. The receptor and the ligand now are collectively called the endosome. And the endosome is typically going to interact with, again, a lysosome or a Golgi so that there can be processing. So we have lysosomes, not shown in this diagram, but lysosomes and Golgi that are typically going to interact with the endosome and they're going to do the processing. And so it's listed here, but they're not shown in the diagram. So the endosome with the ligand is going to interact with the Golgi. It's going to interact with the lysosome, and we're going to have processing of what we brought into the cell. You'll notice that the receptors have separated. So the receptor has separated from what you brought into the cell. We go off for processing this way. The receptors can be recycled, though, back to the plasma membrane. So we basically use a transport vesicle with the receptors inside of it, and we move it back to the plasma membrane. What you're really looking at here in this form of recycling, so this is really just membrane recycling. Okay? This recycling is really just a form of exocytosis. What's not happening, though, is you're not releasing the receptors to the outside, right? The receptors aren't floating around in the extracellular fluid. What's happening is you're merging the membrane, right, the vesicle membrane, back to the original plasma membrane. So we're merging these two together here. And then you're reinserting those receptors. We'll put R here for receptors. Right? We're reinserting those receptors back into the plasma membrane so they can bind to ligand again. So we'll put an L for ligand. Okay? So those ligands now can interact back with the receptors, and you can start the process all over again. So this diagram actually shows you receptor-mediated endocytosis, but it also shows you exocytosis in action as we recycle the receptors back to the plasma membrane. In the process of exocytosis, you're really just talking about a reverse of endocytosis. So cells use exocytosis to reincorporate receptors back into the membranes. They use exocytosis to put specific proteins, put channel proteins and carrier proteins in the plasma membrane. But we also use exocytosis to get rid of things as well. You can exocytose proteins into the extracellular fluid. You can get rid of cellular waste through exocytosis. So there's a lot of uses of the exocytosis process, release neurotransmitters, hormones, etc. Exocytosis utilizes proteins, the RABs and the snare proteins. RAB proteins are basically docking type proteins. Snare proteins actually are involved in the fusion mechanism. And we'll look more at that when we get to the nervous system and we talk about exocytosis of neurotransmitters. The interesting thing to note, again, about these processes is that they require energy. Right? ATP is necessary for endocytosis and exocytosis. Turns out we're even going to see that calcium is involved in the exocytosis process. Goblet cells use exocytosis to release mucus. Fibroblast cells use exocytosis to release proteins into the extracellular matrix. So there's lots of different examples. Neurons for neurotransmitters, endocrine cells for hormones. So lots of examples of exocytosis taking place in the human body. The last thing I want to go over with you guys is just kind of putting transport all together. So when you look at transport across epithelial cells, there's a lot of different transports to consider. We've been looking at the idea of utilizing, channel proteins and carrier proteins. So on this diagram, you can see some proteins embedded in the membrane. Okay, and so we're talking about this idea of transport across the cells. But we typically use vocabulary to describe that transport. So we use words like absorption. So when we move molecules or ions from the outside to the inside of the body, there's this absorption that's taking place, right? Moving things across the cell. And we're going to utilize channels and carrier proteins to do that. We also have a term called secretion. Secretion is when we move things out of the body. So when we're secreting, for example, when we're secreting from the extracellular fluid and putting it into, say, the nephron system. And if we secrete into the nephron, then we're allowing for excretion to take place. So absorption going one way, secretion going another direction. Transcellular means to move across the cell. So these black lines represent transcellular, right, moving across the cell. Paracellular means to move between the cells. So there's different terminology that we oftentimes run into when we're trying to describe different types of movement across the cells. And then when you look at sort of the big picture of transport, a great example is this transcellular transport of glucose across epithelial cells. So just in this one example, we can see a glut transporter, and a glut transporter uses facilitated diffusion. We can see a primary active transporter. This is the sodium-potassium pump. And since this carrier protein is tied to the primary, this would be a secondary active transporter. So here we have sodium and glucose, let's say moving from your digestive lumen, and they're moving into the cell. Sodium is being moved out by the sodium-potassium pump, and then here we have the glucose moving by facilitated diffusion to get into the extracellular fluid, so into your bloodstream. So it's the big picture of transport, just looking at how glucose might move from your digestive system into your blood. Why is this of interest to us? I'll give you a clinical usage here, oral rehydration therapy. When we mix salts and sugars and water together in solutions like a Pedialyte or a Gatorade, what we're doing is we are enhancing the transport of water across the epithelial cells. So as you move the salts and the sugars across the epithelial cells, you can draw water with it. So water is going to be absorbed across the epithelial cells better when you're also absorbing sugars and salts. So to assist in rehydration through an oral capacity as opposed to an IV, right? IV is intravenous. That's where we plug directly into a vein. Okay? But if we were just trying to rehydrate a patient by them drinking fluids, drinking water is good, right? Drinking water is good, but drinking water mixed with salts, right, like sodium chloride and sugars, okay, is better, because we can see that the transport of those salts and sugars will also begin to draw water across those epithelial cells. So that's the end of transport. I hope you guys enjoyed that. Take care.